All right, hello biology people. I'm back to continue our discussion on cells. So up to this point, you should be knowledgeable on the life qualities. We talk about growth, development, reproduction, being able to maintain homeostasis, metabolism, growth, development, the species being able to adapt over evolutionary time, um, all the aspects of life, right? So all packaged within one little plasma membrane, and we call that then the cell. We started out with version 1.0, right? The prokaryotic cell, bacterial cells, with only four minimal qualities: the membrane, our cytoplasm, our nucleic acids, and our ribosomes, plus all of those life qualities, right? Now we're going to look at version 2.0. Right? So over time, there was necessity for more complexity, right? And we start to have this greater complexity inside of a larger cell. And we're going to call this then a eukaryotic cell. Eukaryotic is going to be sort of defined as having a membrane enclosed nucleus and other organelles. So these are the organelles that are absent in the prokaryotic cells, the organelles that are now present in the eukaryotic cells. And we use the term organelles kind of like analogous to organs in the body. So the heart, the brain, the stomach, right? The liver, the kidneys. These are like little compartments inside of our body. And each one of these compartments has their own particular job, their specialization that they do. And then they coordinate, collaborate with the rest of the organs in the body, and then we have these functional systems. So it's the same idea inside of a cell. We're going to have little specialized little compartments, specialized organelles that all collaborate together to then allow that cell to function properly. And the types of organisms that have these eukaryotic cells are going to be things like pond water creatures, the protists. We have mushrooms and fungi, plants, and animals. And again, taxonomically speaking, humans are animals. So we ourselves are also comprised of these eukaryotic cells. So just giving us a size reference, here we have a smaller prokaryotic cell. And relatively speaking, we look at this sort of idea of a eukaryotic cell. And you can imagine how much bigger that's going to extend to the left. Up and around, and then, large, right? And we see these little compartments, these organelles inside of that eukaryotic cell that we don't have inside of the prokaryotic cell. This is an artist rendition of a eukaryotic cell. Very colorful, and in life they're not this colorful, but for teaching purposes, we make very clear color distinctions on the different organelles. Right? This is that large purple organelle that we would then define as the dominant. That's where we get the name eukaryotic cell, right? True nucleus, right? There's a nucleus. And we have these little blue folds, this sort of pinkish little sort of membranous folds, orange. Again, all of these little compartments within that plasma membrane. So there's our plasma membrane. And that becomes then that plasma membrane, that outer sort of boundary. And this cell with all this organelles floating around in that cytoplasm, we then call a eukaryotic cell. And this is an example of an animal cell. Right? An animal cell is going to look different than a plant cell. But again, plant cells are still eukaryotic. So animals are eukaryotic. Plant cells are eukaryotic, but there's distinctions between the two, just like we're different from the plant. Everybody knows we're different from the plant, but there are some similarities, but obviously there's some important differences. First of all, we have a very rigid boundary outside, external to that plasma membrane. We have what we call a cell wall. We have these little green organelles that are not visible in the animal cell, not present in the animal cell. We have this large clear compartment that is not present in the animal cell. But we do have the nucleus, these little blue folds, pink folds, orange folds. So again, similarities, but still differences between plant and animals, both eukaryotic cell, but different specializations, different organelles found in each cell type there. So let's start off with the nucleus, right? This is where, again, we get the name eukaryotic cell, having or containing a nucleus. If we use the analogy of organs in the body, right, the nucleus would basically be the brain of the cell. This is what controls the human body. Right? We need the heart to beat, we need the kidneys to filter, we need the liver to digest and all that, send all these digestive enzymes and detoxify. But if the brain shuts down, right, that shuts down the entire system, right? The heart can keep beating if the brain is gone, but again, there's no consciousness at that point, right? So just we see the brain is a very, very vital organ. The nucleus is a very, very vital organelle. It contains all of the cell's DNA. It's going to control the cell's activity, and it's going to then be protected, or I should say, there's the DNA inside that nucleus is going to be protected by a phospholipid bilayer inside of a second phospholipid bilayer. Right? So it's kind of hard to visualize maybe, but we basically have a hydrophilic, hydrophobic, hydrophobic, hydrophilic, and then again, hydrophilic, hydrophobic, hydrophobic, hydrophilic. So it's like two membranes sort of folded around each other. So it's basically a membrane within a membrane within that larger membrane that would be the plasma membrane. So a lot of membranes here, right? So inside here we have DNA, we have our first membrane, second nuclear membrane, and then this blue liquid basically would be the cytoplasm. And again, inside there we have our DNA, and we have these pores, right? These nuclear pores. I'll give you a second there to think, well, why would we need pores? What has to exit the nucleus? Right? Well, basically we have an important molecule that we know of as RNA, right? You remember we transcribe DNA into RNA. DNA is big. It's not designed to move. It's a double helix. RNA is single strand. It's designed to send messages to different places. So RNA would be generated in here, but then RNA has to leave the nucleus through those nuclear pores. They're basically modified IMPs. They're basically always open, right? So they're exit ports for that RNA. So again, different picture, but we see all of our DNA. We see this purple nuclear sort of double membrane, nuclear envelope, and then these pores. So pores are important. They allow RNA out, but they have a sort of a flip side that basically is not good, right? Just imagine that your front door was always open. And it's very easy to go out. And you don't have to open the door, you just can walk out. If your window was always open, no screen or nothing, you could just go out, right? But if we have these things always open, there's a potential for something to come in. And again, why do humans have to cope with viruses? Like right now, the COVID, right? Why do people have to have a threat of getting a viral infection? Well, it's because of the physical design of this nucleus. Just like RNA can go out, viral RNA or viral DNA can easily come into that nucleus and start to damage our DNA. So it's a flaw in the system, but hey, it seems to work. We're healthy most of the time. And again, there's got to be movement across that boundary. RNA has to leave out. But again, our first very important organelle being the nucleus. A second important organelle would be the endoplasmic reticulum, or for short, we call it ER. If you're in the medical field, ER means emergency room. If you're a cellular biologist, ER means endoplasmic reticulum. These are basically folds and folds and folds and folds of membranes. And what they then accomplish is they provide a physical location for the formation of different macromolecules, right? So think of them as factories, right? This is where we're going to make and modify carbs and lipids and proteins. But we have different types of factories, right? We're going to have what we call the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So the smooth, the surface is very smooth, right? You can kind of see this idea of very smooth. Real life is kind of hard to tell what's going on, but we have smooth outer surface. And this smooth ER, this is going to be a factory that makes and modifies carbohydrates and lipids. So the body can manufacture these molecules if necessary. If you're not supplying it with enough, the body can start to make, we can make steroids, we make cholesterol, we can, repair things that have become damaged. So important organelle, the smooth ER. The rough ER is going to have these little like bumps on it, right? I don't know if you can see how bumpy that looks. Bumpy, 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 these little black, little sort of like chocolate chip looking things. Those little black specks are actually ribosomes. And ribosomes then are going to be surrounding that whole environment. And we know that ribosomes are important in the formation of proteins. So if you have ribosomes on your surface and you're a factory, it's sort of logical to say, well, this factory isn't going to be associated with making proteins. So we make and modify proteins within the rough ER because of the ribosomes that are present. So let me back up a couple slides. So in the animal cell, again, we have our nucleus. So DNA transcribing to RNA, then RNA leaves out and right away bumps into our endoplasmic reticulum. It surrounds in 3 dimensions all the way around that nucleus. Again, some parts being smooth, some parts being rough, so we can make different types of products. And the plant cell is the same thing. Right? We have nucleus, and then right adjacent we have our ER, rough and smooth endoplasmic reticulum. So again, important. So we have then this boss molecule that's sending out instructions to make products, and then we have the factories that are making those products, right? The carbs, the lipids, and the proteins. Now we have another important organelle that is called the Golgi apparatus. This is like the Federal Express, the Amazon, the shipping, the postal service of the cell. Once the ER makes the product then has to be distributed either within the cell intracellularly or outside the cell extracellularly, right? So the Golgi apparatus is going to package and transport molecules again within the cell or to the membrane so they can be distributed out of the cell. So we're going to receive the product from one side and then ship it out the other side. Like when you go to the post office, right? You drop it off the front door, and then the back door, it gets distributed, goes to where it needs to go. So we have then now 3 important individual organelles that are working together, collaborating together, and by definition, we then have what we call a system. So we have like in the body, we have organ systems. Here we have organelle systems, right? The nucleus sending out the instruction, the endoplasmic reticulum making the product, and then the Golgi apparatus distributing the product. Right? So these 3, the nucleus, ER, and Golgi, comprising our endomembrane system. And again, we can transport internally, or we can start to transport externally outside of the cell. So important. All right, so I'm going to stop there. The next organelle gets a little bit involved. So let me stop here and get this video transferred here.